Hello. So let's look at the week 2 chapters. Remember, all of this loveliness is going to be in the week 2 questions thread under my notes. So that will be a good, easy review for you. I'm going to put it on page width just to make my life easier. Let's start with chapter 8, of course. So definition. This is all going to be about cellular replication. First of all, cell division is when a cell splits into two cells that are identical, the old cells, so the original ones, the parent, and this is not sexual reproduction because they are identical, right? Chromosomes, these are chunks of DNA and genes. The number varies with animal, the number of chromosomes, like chromosome 1, chromosome 2, chromosome 3, that's what we call autosomal. X, Y, whatever, if you're with birds, it's called Z. Those are the sex chromosomes. In humans, like I said, we are X and Y's. See, I even put Z in birds. I just thought that was funny. All right, so asexual reproduction. This is when your critters, your organisms, reproduce by cellular division, cell division. There's no sex, no eggs, no sperm. The kid is the same as the parent. We can call this a clone. If you want to talk about why this might be bad, check out the great little video. Sexual reproduction, a little different. This requires the fertilization of an egg by a sperm in humans, of course. The production of the egg and sperm cells involves a special type of cell division called meiosis. So the previous one is mitosis. This one is meiosis, which occurs only in reproductive organs, such as testes and ovaries in humans. It allows for more variation, which in turn allows for more evolution, for evolution to work on. Basically, because you're not a clone, you're going to be different than every other critter of your kind, which means you have a lot more variety. This could be really good. And there's a good little link. Let's go down. Just for fun, this is the number of chromosomes in body cells. So you see Indian muskrat deer have 6, koalas have 16, giraffes have 30, humans we have 46, and this is numbers, so there'll be 23 pairs. And duck-billed platypus, which is a monotreme, so before placental, has 54. So it does not matter how quote-unquote evolved you are. And the best is this red viscacha rat has 102. It's amazing. Okay. And then also, obviously, chromosome does not correspond to size because our little rat has way more than the humans or the buffalo. Let's go down. Alright, so here's the general phases. Right, most of our time our cell's in interphase, or just kind of hangs out and grows and doesn't do too much, just its normal everyday duties. And once in a while it goes through the mitosis or the meiotic stage. That's only about 10% of the time. That's when it produces a clone. In this interphase, which is most of the time, it goes through the S phase, that's where it duplicates any chromosomes. G1 phase, which is regular cell function. G2 phase, preparing to divide. Then we also have the mitotic phase. Okay, this book lumps them all together. The mitotic phase is where the cell separates. Mitosis is the process by which the cell gets the exact number of chromosomes. And cytokinesis is where everything else gets shoved into each cell beyond the DNA, right? There's a little checkpoint, test you out. So let's look at mitosis in all its glory. So interphase, right, we just kind of hang around, do what we want to. Cell growth, blah, blah, blah. These are little, you see the little centrosomes? Those are hanging out, not doing much. We got our chromatin is all bubbly and looks like spaghetti. And then you got our nuclear envelope and our plasma membrane. Now, once we get over here, let me scroll down so you can see the name. Over here in prophase, we're going to get our chromosomes now turned up into their little pairs with their centromeres. Our centrosomes now have some spindles. And then it continues on, and our nuclear envelope is going to break up. Our centromeres have moved to polar ends, right? And now we do have the spindles. So let's go down to the next one is metaphase. So metaphase is where all our chromosomes line up in the center, right? Anaphase chromosome pair, one chromosome goes to one side, one chromosome goes to the other side. Telophase and cytokinesis. So now we got all our dudes on each side. We're reforming our nucleus. We've each got some little chromatin over here. Not chromatin, centromere. And then our cleavage furrow. See the little indentation? And that's going to form a plasma membrane between the two cells. So we'll have a nice little copy. Slightly different in plants than animals. Animals, it goes, I believe, out to in. Plants, it kind of goes in to out. See if they have an example. Briefly, so prophase, as we saw, chromosomes condense, nuclear membrane goes away. Metaphase, sister chromosomes pairs line up in the center. Anaphase, sister chromosomes part ways. One set goes to each side of the cell. Anaphase, telophase, and cytokinesis. Nuclear membrane forms around each set of chromosomes. Cleavage furrow forms and leads to two separate cells. Oh, here we're going to do animal and plant. See, in the plant, it starts on the inside and goes out. Animal, it does it the other way. Just because one has a cell wall. So pretty. That was a good video. Another little checkpoint. It's fun. In embryo, birds develop teeth. Let's just think about the development. If you look at embryonic development, especially for humans or other species, you can kind of see their evolutionary past appear and then disappear. Some fun videos. Cell cycle control system. So this is specialized proteins within the cell. They integrate information from the environment and from other body cells to send, stop, and go-ahead signals at certain key points during the cell cycle using signal transduction pathways. Remember our signal transduction pathways? This is basically saying like, hey, go ahead, do the next stage, hey, don't, hey, hang out. The tumor is caused when cells grow to excess, right? So they kind of think they're still babies and they're just going crazy and growing, and their cell cycle control system isn't stopping it. What is a benign tumor? A benign tumor is abnormal cells that remain at the original site, and they cause problems if they grow too large and disrupt certain organs, such as the brain, because you don't want your brain messed up. But often they can be completely removed by surgery. Think of any time of people saying something like a growth in the stomach, right? I said, I wonder the issues when it happened to women historically and doctors could not examine women, etc., right? And then a malignant tumor, this is different than benign. So benign, you can probably just chop the sucker out. With malignant, we have cells that spread into neighboring tissues and other parts of the body, displacing normal tissue and interrupting organ function. This is typically called cancer, and so it's bad because you can't just chop out one chunk. Radiation therapy burns out the cancer. It does damage some healthy cells, right? We're doing a lot of different research in these areas to try to find a way to specifically target cancer cells. Chemotherapy poisons by stopping various parts of mitosis. There's another checkpoint for you to review. So what is a somatic cell? Did I go into another chapter? Nope, just a whole different section. Okay, so somatic cell is an everyday cell in humans. We have 46 chromosomes and 23 pairs, remember? A karyotype is a picture of matching chromosome pairs shown by increasing numbers. You'll often see this when they're talking about like Down syndrome because they have the extra chromosome pair, extra chromosome set, so they have 3, 19? Something like that. We'll talk about that later, and you can be like, oh, Heather can remember. Gametes are the egg or the sperm. So those are very specific. So most of your cells, we just call them somatic, and then gametes are the sex cells. Homologous chromosomes, these are 2 chromosomes of such a matching pair. So it's a matching set, and they carry genes controlling the same inherited characteristics, right? There's a fun little sock experiment that really worked well for my understanding of this. Sex chromosomes determine a person's sex, male versus female. Remember, females have 2 X's, and males have an X and a Y. Some links that are fun Autosomes, so those are all the chromosomes except for X or Y, are all the number chromosomes. So chromosome 1, for us, it's going to be 1 through 23. You get one set from your dad and one set from your mom. Diploid means all cells except the gametes have a pair of chromosomes. So gametes, right, I'm only giving, if I as a woman would have an egg, it would have half of my chromosomes, so it would only have 23 out of the 46. And then if it was fertilized by a sperm, the sperm would have the other 23 out of 46, and together we would get a complete set. But normally, the rest of your body already have the 46. The total number of chromosomes is 46 in humans. It's a diploid number. This is represented 2N. And thus, a triploid would be represented as 3N. If you had a quadloid, 4N. Haploid means one set of chromosomes, right? So our somatic chromosomes are diploids. Our gametes are haploid. This is gametes. This is just called N. In humans, or whatever, it's just called N. Any other weird ways to get kids? Think of ferns. So is this the only way for sexual reproduction to occur? Probably not. Feel free to do some research. It's quite interesting. So how does meiosis work? So interphase before meiosis, everybody pairs up, you get your duplicates, right? In meiosis 1, you're going to have your homologous dude split up. In meiosis 2, you now break the pair up. So first, like, say I have 2 at number 2. Let's say number 3. So our 3 at our, we duplicate our chromosomes, right? So this is chromosome number 3. Now we're going to have 2 sets of chromosome number 3. Now in meiosis 1, I have 1 pair and 1 pair. This is very similar to mitosis, right? But in meiosis 2, now I got only 1 set for each little gamete. So the meiosis is the process that produces haploid daughters and cells in diploid organisms. This resembles mitosis for the first section, but has some special features. So what are those special features? I wonder. All right, let's look at meiosis a little bit closer. So interphase looks kind of the same as it did for mitosis. Then in prophase, we're going to, get into less spaghetti and more paired looking. Our nucleus again disappears. But here we have a special thing. This is crossing over. You see the chromosome pairs, they don't want to be exactly like your mom and dad originally gave you, so they swap sections just to be friendly. Then in metaphase, we're going to line up. Anaphase, we split. This was all for meiosis 1. So let's talk about what happened in that first section a little bit more. So meiosis 1, results in 2 daughter cells. We saw that. Prophase, prophase 1, the chromosome pairs, one from your dad, one from your mom originally, split up. Nuclear membranes say goodbye, crossing over. So that's what keeps the mom and the dad from being the same as they originally were. Metaphase, everybody lines up in the center. Anaphase, pairs separate, but the sisters remain together. Telophase, 2 daughter cells of haploid set, right? And we still have sister chromosomes. So let's go down and look at meiosis 2. So we have 2. That already happened, right? So we're going to do our prophase. So nucleus never actually reformed, but we get our fun little doodads to hook up to. We're going to have metaphase 2. We're back up together. See how the crossing over has occurred. There's a little blue on the red, a little red on the blue. Anaphase, now we're going to break up our sister chromatids. And then in telophase and cytokinesis, we're going to have haploid daughter cells created. So we finally got down to 1 set. Let's go down. Let's read the review. I have a miniature pincher who wants love very badly. So meiosis 2 is very similar to mitosis. Prophase 2 is back to nuclear membrane breaks. Wasn't really formed, but it might be there for a second. Metaphase 2, the sister chromosomes line up in the center. That's what we do in metaphase. In anaphase, we split. But now chromatins, the little chunks of chromosome, go to each side. And then in telophase 2, each of the daughter cell